Okay, so I am doing this in a slightly different format. Unfortunately, over this holiday weekend, they ended up changing up the screen capture programs on our computers. And so, anyway, for now, I'm going to use WebEx. It's my hack until the help desk is able to give me the administrative rights to put on the new screen capture program. So I apologize. This one's probably a little bit of a rougher video than normally what I would make. I also want to apologize because right now, I'm just getting back from some field work, so I'm in my more casual attire here, hence the hat, but better than my hat head. So anyway, this is going to be a video on the muscle and nervous tissue, my intro video for histology. So actually, I'm going to go ahead and swap up. I just want to make sure that the screen capture is getting the appropriate thing. So I'm going to make sure that you're getting the PowerPoint this time. So let me go ahead and dive right into this. Again, this is just going to be the introduction to muscle tissue and nervous tissue, which is going to be your last lab for histology this week before we get into the skin or integumentary system. So we haven't talked about muscle tissue yet. So remember, again, there's those 4 general tissue types, right? We have epithelial tissue, connective tissue. Now we're getting into muscle tissue, and then we'll do nervous last. So again, the general tissue type is going to be muscle tissue. And when we talk about muscle tissue, again, obviously you probably kind of know this is going to contain contractile fibers. So these are going to be fibers, again, that are going to be modified for their ability to be able to contract and provide motion. Also just maintenance of posture. And we'll talk about, once we get into lecture, how muscle contraction also produces heat. So these cells are special because, again, these cells need to be excitable and they need to be conductive, meaning we need to be able to excite those cells and then cause them to contract. So the 3 types of muscle tissue, and we'll talk about cardiac tissue. Cardiac muscle tissue has to be able to conduct, which means that from cell to cell, one cell has to be able to pass on the message for the next cell to contract, so they can all contract as one. So anyway, we'll get to that in just a minute. But the 3 specific tissue types, right? So again, muscle tissue is the general tissue type. The 3 specific muscle tissue types that you need to know are skeletal muscle, cardiac muscle, and smooth muscle. Or you'll also hear it called visceral muscle, because viscera means organs. So if you don't know that the term viscera means organs, here's my little hack for you. So when I teach marine biology, I always have a student or 2 that asks me if I like to eat snail or if I like to eat sea urchins. And I always tell them no, because I don't eat visceral mass. Like, I'm just not knocking it. For those of you that do like to eat visceral mass and organs, they, again, also oftentimes have a lot of nutrients in them. But I am not a visceral mass type of person. So maybe that'll help you to remember whether or not you like to eat visceral mass. Do you like to eat organs, or are you like, no, I primarily like to stick more to the muscles and not the internal organs. So that's, again, my kind of trick for you. So when we talk about smooth muscle, or oftentimes, again, also called visceral muscle, this is going to be muscle in organs. So again, like your digestive tract is going to be lined with smooth muscle. And that smooth muscle, again, is even like the stomach. And that's because you got to be able to push food down that digestive tract, and the stomach has to be able to grind it. And so that hopefully will help you kind of remember where you'll find smooth muscle as well. Cardiac muscle is probably easy. You can, it's heart muscle, so cardiac heart. And then skeletal muscle, again, you're going to find your skeletal muscles. So those muscles that are actually moving the skeleton. So let's go ahead. Oops, I just jumped way ahead here. And let's start to talk about the very first specific muscle tissue type, which is skeletal muscle. So when you look at these cells, they are very easy to identify. One right away, you'll see there are these really long cylindrical cells. So again, a lot of these muscle cells are as long as the muscle is. So there are these really long muscles, or these really long cells, sorry. And so this is what's called a longitudinal section. So here, if you see, here's your muscle, or again, if I take my pen, here's my muscle, and it's like I cut this way. I did a longitudinal section, and I'm looking into that marker. If I cut it in half, okay, I'm looking into it. So the other easy thing to identify in skeletal muscle is that it's multinucleate, which means that it has many nuclei per one cell. So you can see here, you know, this one has many nuclei per this one skeletal muscle cell. So that's another way to identify. And the nuclei are always going to be on the periphery, just like you saw in your adipose cells, right? Because the adipose tissue, the lipid droplet took up all of the inside and it pushed everything out towards the outside. So again, here you're going to see something similar with your skeletal muscles, right? We have all of the contractile fibers in the center of the cell. And then it's going to push the nuclei out towards the periphery or the outside of the cell. Okay, so that's where you're going to see the nuclei. You're going to see those visible light and dark bands. We call that those striations. So it will look striated. And we'll talk about that when we get into the skeletal, the muscular system. And we'll talk about those contractile fibers and why they form those striations. Also, again, this is under voluntary control. This is kind of more lecture stuff. But again, voluntary conscious control, you control your skeletal muscles, very different than smooth muscle and cardiac muscles. And obviously your skeletal, where you're going to find your skeletal muscle is it's going to be attached to your bones, right? It's going to connect one bone to kind of go from one bone to another so that you can move those bones, and it's going to be connected by tendons. So muscles connect to the bones via tendons, and again, it will provide locomotion of your skeletal system. So again, you know, when you're contracting things like your biceps, you're able to move your forearm, right? Okay, you know, closer to your shoulder and then away, and we'll talk about. We're going to get into that and what those actual terms are, flexion and extension and all that when we get into the muscular system. So that's coming. Fun stuff, though. Okay. So really quick, I need to explain to you, and we'll get into this when we get into the muscular system, but I need to explain it now because we're going to look at some cross sections of entire muscles. And so you need to see how the muscle cells are bundled together. So the first thing is, and this is where it gets a little bit tricky, is we have a lot of our skeletal, remember we talked about there's these contractile fibers inside. So inside a single cell, and this right here, all of this is inside one cell. You can see the nucleus here, and then this would be the plasma membrane that we call in muscle cells the sarcolemma. So this sarcolemma is basically kind of it's actually some connective tissue that's covering the plasma membrane of your muscle cell, okay? So all of this is in one single muscle cell that we call a muscle fiber. I don't know why they call it a muscle fiber because it is very confusing for students, because students are like, it's called a fiber. It makes me think that it's just like a filament, right? But so there's myofibrils, which are the filaments that are grouped together in a bundle. And then all of these myofibrils make up the very kind of meat portion of your muscle cell, which is called a myofibril. So again, I know that that is, or I'm sorry, muscle fiber. I said, and see, I even said myofibril. So your myofibrils are basically your bundles of muscle filaments, and all of the myofibrils will get grouped together in a single muscle cell, okay, like you see here, with again some of the organelles like the sarcoplasmic reticulum, which is basically the endoplasmic reticulum of a muscle cell. And all those will get bundled together inside a single muscle fiber or muscle cell. Okay, so I know I didn't, please don't hate me. I did not make up the term muscle fiber equals muscle cell, but you have to know because the book is going to use it, and in anatomy in general, they'll use the term muscle fiber to refer to a muscle cell. So make sure that you know that muscle fiber equals muscle cell. So again, one of the ways, if you're going to kind of, it's a good idea to do it now because we're going to get to it in muscles anyway. Make yourself a note card and go, okay, you know, you can put that myofibrils is a bundle of filaments. Okay, so put myofibrils equals bundle of filaments, and those group together to make up a muscle fiber, or in parentheses cell, muscle cell. And then you can group together all of these muscle cells, again, muscle fibers, right? Muscle fibers, muscle cells. So here's a muscle cell, here's a muscle cell, here's one, here's another muscle fiber, muscle cell, another muscle fiber, muscle cell here. And then when we group those together, okay, we're going to group those together in what's called a fascicle, okay? So here's your fascicle. So basically a fascicle is a bundle of muscle fibers or muscle cells together, okay? And then we can bundle all these fascicles. So here's a bundle of fascicles, here's a bundle of fascicles, here's another one, here's another one, here's another one. And we bundle all those together into the whole muscle, okay? So when you look at muscles, muscles, again, are very organized. They have these groups of, again, these myofibrils grouped together into these muscle fibers and muscle cells, and then those are grouped together in fascicles, and those are grouped again together to make up your muscle. And so that ensures that you have all of these fibers running in one direction, these contractile fibers running in one direction, all working together for the common purpose of contracting that muscle. Okay, so now here is where it gets a little bit tricky, is for the muscle tissues, we want you to know some certain features in them. So for example, we want you to know that this plasma membrane here surrounding the group of myofibrils, okay, is called the sarcolemma. So it's just some connective tissue. It's covering, again, each of these individual muscle fibers. And the sarcolemma is the equivalent of the plasma membrane for a muscle fiber or muscle cell, okay? Now, when we bundle these muscle cells together, okay, so you're going to see we're bundling all these muscle cells together to connect this muscle tissue to, I'm sorry, muscle fiber, muscle cell, okay? So to connect this muscle cell right here to this muscle cell, there's some connective tissue in between, okay? And that connective tissue that surrounds each of these muscle cells is called the endomysium. So on your note card where you make this, what I would do is I would put right here, in between here, I would put endomysium, okay? And this is the word endomysium. So what that means is that endomysium is going to surround each of those individual muscle cells, okay, to help connect them to the next muscle cell, okay, that will eventually be bundled into a fascicle. Now, here you're going to see these fascicles are going to have connective tissue around them as well so that they can connect to the next fascicle. And that is going to be called, and I would, again, put that up here to remind yourself that, again, those fascicles are going to connect to each other via their paramycium. So the paramycium, you can put that right here, is going to surround each of those fascicles. So peri means around, so it's going around each of those fascicles. And then you're going to, again, those fascicles will bundle together to make up a muscle. And what's the connective tissue that surrounds the entire muscle and is really, again, holding all these fascicles together in that muscle. And that is going to be the epimysium. Okay. And you can think of epi like epidermis. It's the outermost layer. Okay. So epi is going to be on top, right? Okay. So that's gonna be the layers, and I would put epimysium up here to remind yourself that it surrounds the entire muscle. That's the connective tissue that surrounds the muscle, the entire muscle. Okay. And so hopefully that will help you to kind of remember some of these as you get into looking at the cross sections of these muscles. So here you can see this is a skeletal muscle. It's a cross section. Here, this is basically like, again, you had a muscle cell, and now this time you cut it. Oops, sorry, if I can get my finger in the right spot. You cut the muscle like this, and now you turned it this way, and you're looking into the muscle, okay? And so here you can see that cross section of the muscle right here. You're looking into it. And so what you're looking at is you're going to be looking at a bunch of fascicles, right? First, you're going to see all these fascicles that are bundled together to make up the skeletal muscle. And remember, the epimysium would be around the whole entire muscle, kind of holding it all together. And then each of these fascicles, and the fascicles and cross sections aren't going to be these pretty, beautiful round fascicles. Again, this is a prepared slide of a muscle. So it's not going to look pretty, but you can really see how that would be a bundle right here. And we're going to make it obvious, right? So here would be a bundle called a fascicle. So there's a bundle of muscle fibers or muscle cells bundled together in a fascicle. Again, you know, here would be another fascicle, probably right here, and then here. Here's another fascicle right here. Here's another fascicle right here. So again, we're going to make it pretty obvious that you can see it's bundled. Okay. And so, of course, then if I have an arrow here, what are we pointing to that's going around the fascicle? That's going to be the paramycium that's surrounding that connective tissue surrounding each fascicle. Okay. And allowing it to connect to the fascicle next to it. So hopefully that makes sense there. Now, as you zoom in, so again, this is at 50 times magnification. We're going to zoom in even closer. And here we're going to zoom in, and you see here's part of that paramycium going around this big fascicle. And you can't even see the whole fascicle now, right? You're zoomed in on, here's just this one big fascicle. And you can see inside that fascicle here is going to be all of the skeletal muscle cells, or again, you can call them muscle fibers, or you can call them myofibers. So myofiber equals muscle cell equals muscle fiber. Whichever of those that you want to use on the practical, to be honest, I will give you either, as long as you know that it is each of these is an individual muscle cell, okay, or myofiber. So again, these myofibers, not myofibril, because those are the little, again, filaments inside the muscle cell, but myofiber. I know, please don't hate me. I did not come up with these terms. I know they could not make them more confusing. So I'll try to always say, if I use the term myofiber, because sometimes it's just ingrained in me, I'll always try to say muscle cell as well, to remind you that each of these is a myofiber or muscle cell, okay? Individual muscle cell here and here. And again, these individual muscle cells are all bundled into this big fascicle, right? So now you can look and see here, each of these individual muscle cells is going to have connective tissue surrounding them, surrounding their sarcolemma, right? Which would be the plasma membrane. And you can't see the sarcolemma. I'm not going to tag the sarcolemma on this because look, you can't see it, right? It would just be this little membrane that's covering it. But I could ask you, what is right here, if I'm pointing here, what is this surrounding, what is this connective tissue surrounding the myofiber or the muscle cell? And then you would say, oops, where'd my arrow go? There we go. And so then you would say it's the endomysium, okay? Endo is that's the innermost connective tissue layer that you're going to find, okay? And so the innermost one that you're going to find, you can think endo, inner, is going to be right around each of these muscle cells. Okay, so hopefully this helps you. Again, a little bit, if you're confused, I can always set up some time with you. I always like to have you, wish I had you in lab so I could show you all this. Okay, so again, this is zoomed in even more. So this is at 400 times magnification. So here you're looking at individual muscle cells or myofibers, okay? So again, here's a myofiber, individual muscles. And look, you can actually see the peripheral nuclei, the nuclei kind of shoved up against the sarcolemma or plasma membrane, right? And so you can see all that. And then this connective tissue, again, surrounding the white here, surrounding each of these individual myofiber muscle cells is going to be the endomysium, okay? So that's a really nice slide. So, and again, the sarcolemma is just the term for the plasma membrane of a myofiber. And the reason you're going to ask, well, why don't you just call it plasma membrane like it is, right? You guys are like already overloaded. And the reason is that it is going to have a different function. It's going to have an extra function that allows these cells to be contractile. So that's why we give it the special name sarcolemma, because it's got some extra features in it that we'll get into when we get into the muscular system. Okay? So it's a good idea just to get used to these terms now anyway, because they're coming. So it will really help you once we get to the muscular system. So again, another zoomed in cross section here at 400 times magnification. You can see here's these myofibers or muscle cells, individual muscle cells surrounded by the endomysium. And then we have, again, many of these peripheral nuclei for each of the cells. Okay. Here's a longitudinal section. So again, now we have a muscle that we cut into longitudinal, like I can get my pen this way, okay? And we're looking into it lengthwise. And so you can see each of these here, like here's a myofiber or muscle cell. Here's another muscle cell. Again, these are really long cells, right? Depending on how long the muscle is. And you can see again that they're going to have many nuclei pushed up towards the periphery. And again, in between here, surrounding each of these myofibers is going to be the endomysium, okay? We're zoomed in again here at a longitudinal section, 400 times magnification. So here you can see again our myofiber muscle cell here. You can see the nuclei shoved up against the periphery here. And then you can see they're striated and you can see the endomysium surrounding them, okay? And again, the sarcolemma, I can't remember if we marked the sarcolemma on there, but we may ask you something like, what's the term for the cell membrane of the skeletal muscle? And so it's going to be sarcolemma. So again, good to just know this because you're going to have to know sarcolemma when we get to the muscular system anyway. And it'll just be one less thing that you have to memorize when you get there. Okay. If you have any questions on skeletal muscle, please let me know and I will set up some time with you. So let's jump into cardiac muscle, which is obviously cardiac heart, right? So this is going to be the muscle that makes up your heart, again, muscle tissue. And so your cardiac muscle cells are going to be very branched, which this makes sense because they're going to want to connect to each other so that they, again, the muscle cells all contract together as one. We call that a syncytium. So the cells will be branched, but they're going to have one nuclei, okay? So they're not multinucleate like your skeletal muscle tissue is. So just one nuclei per cell. But the cells are goofy because again, they like, look at this one. This is one cell that has many branches. Okay. And then, so here's another cell that has many branches and connects to the next cell. So then these cells are going to be involuntary, under involuntary control. Okay, so we talked about these ones. We talked about skeletal muscles under voluntary control. You, again, are consciously contracting your muscle, right? You have control of that. You're not sitting there thinking about contracting your heart muscle. Thank goodness, right? I mean, thank goodness that it's under involuntary control, because if I had to remember to, you know, make my heart muscle contract so that my heart would beat, I would not make it very long. So again, thank goodness that it's involuntary and it does it on its own. We don't have to think about it. It is striated, so you can see again those lines here, the dark and light lines. Those are the striations. So again, that has to do with, and we will get into that when we get into the muscular system. It has to do with those contractile fibers that are inside the myofibrils. And then again, these cells are going to have something called intercalated discs. So if you look here, there are going to be these lines here. You can see them. They're really pronounced right here. Here's some intercalated discs. And so this is going to be where one cell is going to connect to the next cell. And you're going to have these, what are called intercalated discs, where you actually have connections like gap junctions between one cell and the next cell. And that's because you want to have, you also have desmosomes holding those cells together so that as they contract, right, they don't pull apart from each other. That would be terrible as the heart's contracting if all of a sudden the muscle cells just pulled apart from each other. Okay. But there are also going to be gap junctions in between here. So that way, then as this cell contracts, it's going to share its cytoplasmic contents with this cell that will cause it to contract. And that will make more sense once we get into how these muscle cells are contracting. And we'll talk about some of the ions that are involved with that. Okay. So those are those intercalated discs. I always think you don't have to know what the intercalated discs are, but I always think it helps to know why we have you know them. And so they're important because they hold those cardiac muscle cells together, and then they also are going to pass ions and allow them to contract together at the same time. Okay. So here's cardiac muscle tissue. You can see, you see lots of these. One of the easiest ways to identify cardiac muscle tissue is look for these intercalated discs, right? It's not, again, you can see if you zoom in, you can see the striations, but the intercalated discs are very different than your just regular pattern striations, okay? So that's one of the first things you're gonna look for. If it's got intercalated discs like this, cardiac muscle tissue right away. Also, the cells are very branched. Look at these cells like kind of branching all over like tree branches. Again, cardiac muscle tissue. And then you can see the nuclei are always, of course, going to be stained dark there. Here's more cardiac muscle tissue. And you say, oh, how would I know this is cardiac muscle tissue? Again, here you can look and it kind of looks like it might be skeletal muscle all squished together, right? But one, these cells aren't these really pretty defined long cells like skeletal muscle, and boom, intercalated discs. Okay, so again, intercalated discs are a key giveaway. It should make it very easy for you to identify this cardiac muscle tissue. Okay. Smooth muscle. So smooth muscle is pretty easy, other than sometimes people get it confused with dense regular connective tissue, and I'll show you the side-by-side slides so you don't mix them up. But smooth muscle is pretty easy because these are these spindle-shaped cells, kind of all squished together, okay? So that's the easy way to identify them. And they have this one single central nuclei that's fairly large, okay? So smooth muscle, again, are going to be these spindle-shaped cells, really large central nuclei. Remember that these muscle cells and this muscle tissue, smooth muscle tissue, will be surrounding your organs. So especially the walls of hollow organs, like your blood vessels, like your entire GI tract, like, again, your stomach, like your bladder. So again, these are going to be surrounding these hollow organs. and again is under involuntary control. Again, thank goodness, because you don't have to think about your esophagus contracting to push that food bolus down, or your stomach contracting to be able to grind up that food in it, right? If we had to think about all those things, then gosh, so you're mean, you got so many other things you'd never remember, right? Like, oh, I forgot to contract my esophagus again. So again, involuntary. And then another big key giveaway is it's not striated. So this is the only muscle tissue that doesn't have any striations. So you don't see those light and dark lines. And that's because these fibers, contractile fibers, are very different. And we actually get into that when we get into the muscular system. So one of the best places to see this is I actually have you go back to the intestine slide, the mouse intestine slide, and you zoom in. And so remember, before, when we looked at the mouse intestine, we were actually looking at simple columnar epithelium, which lines the lumen, right? But if you zoom back on that intestine, remember, again, surrounding your hollow organs of the GI tract is going to be smooth muscle. So you have some connective tissue here, again, connecting that epithelial tissue layer, again, to the outside wall here, which will be that serosa that we talked about, again, with that simple squamous epithelium. So you have lots of different tissue types here. It's a good slide to be able to review all those tissue types. And so in between here is going to be the muscle tissue that actually causes the intestines to contract, right? And so it's smooth muscle. And what's great is you actually have smooth muscle crisscrossing each other, okay, when it surrounds the intestine. So, you know, if here's your intestine here, so you'll have a layer of smooth muscle that's going to be the longitudinal. So here's my intestines. So my inner layer is going to be like this of smooth muscle. Okay, so it can contract this way. And then the next layer on the outside is going to be crisscrossing it like this. Okay, so that's why you're going to see here in this section, you'll have that inner layer looks like a longitudinal section. And then here you have a cross section. And that's because those two muscle tissue layers are crisscrossing each other of that smooth muscle. So they're both smooth muscle. Really obvious to see that it's smooth muscle right here, because you can see these, again, muscle cells are more spindle shaped. They're squished together, and you can see these big nuclei. So let's zoom in here. So again, here is a good, really good example of smooth muscle. You can kind of see that these are spindle shaped right here. See here, you can see it's kind of a spindle shaped muscle cell. But the big thing is, look at how big and stretched out those nuclei are too. Okay, so that's another key giveaway that you're looking at smooth muscle. And then here you can look at the cross section of smooth muscle. So we're not going to give you like only a cross section of the smooth muscle and say, okay, what is this? Because it's too hard. We would give you a longitudinal section in there as well, so that you could see that spindle shape of the muscle. Okay, so don't freak out. I mean, you're like, what in the heck? How am I going to identify this? So, but we could use this to ask you about the endomysium, which endomysium is going to be the connective tissue that surrounds each of the muscle cells or myofibers. So again, you're going to see that here surrounding each of these muscle cells, smooth muscle cells. Okay, so you'll still have the endomysium surrounding them. Okay, and so because I put this in here, because a lot of people are like, how do I differentiate dense regular connective tissue from smooth muscle? Because they look kind of alike. But when you put them side by side, you're like, oh, no, they don't look anything like each other. Okay, so that's why I put them side by side here. So look here, when you look at your connective tissue here, the dense regular connective tissue, all these collagen kind of fibers are kind of wavy. They kind of almost look hair-like, right? And it's going to look more hair-like than your smooth muscle is going to. Also, look at the nuclei are usually stained purple, and they're these tiny nuclei. They look like these little like purple freckles, right? Throughout. So that's another way that you know that that's dense regular connective tissue. And here, look at these. Look at these large central nuclei. Again, smooth muscle, they have large nuclei. Little tiny nuclei. large nuclei, okay? So that's another way that you can identify it's smooth muscle. So hopefully that will help you out in terms of identifying those two from each other. Okay, so that's it for that slideshow. I'm going to go ahead and cross over here and share another screen with you. So I want to share actually this screen. Hopefully this should work out for me. And I want to go ahead and go back and share. Let me just minimize this. So again, hopefully you kind of know where you're going to find all of this. So I'm just going to click return to histology so that you can see where you're going to find all of this in the lab. So again, remember, you're going to go to that histology lab, and we're on this last one, muscle and nervous tissue. And here you can see it'll list out some of the main features that you need to be able to identify. And it'll talk about, again, the things like nuclei, striation, sarcolemma, endomysium, paramysium, fascicles, epimysium, all those things that we talked about. Yeah, we covered them all, I think, actually. And I even talked about the desmosomes and gap junctions, which you can't see. Again, we only bring that up in the intercalated discs so that it kind of clues you in when we get into the muscular system as to why we have those. So again, I just always like to give you that explanation. Why do we have intercalated discs? It's hard to learn about intercalated discs without knowing what they do. Okay, so that was all the cardiac, all the muscle tissue stuff. I had cardiac in the brain. And again, you can get to that PowerPoint slide here. Here is the slides for the unlabeled line, leader lined, and the lined and labeled muscle tissue. And then we're going to go to the images of the muscle models. So I want to go ahead and show you really quickly, briefly, because I forgot to show you the models last time. And sometimes we do use the models on exams. They're nice and easy to show things like intercalated discs. So again, if you got this muscle model and you're like, I don't know what this is, okay? Ask yourself, does it have striations? If it has striations, it's either skeletal or cardiac, okay? So it does have striations here, these kind of regular interval lines, okay? So it's striated. So now we've narrowed it down to cardiac skeletal. It's not smooth because smooth doesn't have striations, okay? So then we're like, oh, it has intercalated discs, these random, really dark lines showing that they're the intercalated discs. And it only has one nuclei in the cell. Again, remember, skeletal muscle is going to have the peripheral nuclei, and it's going to have many of them. It's called multinucleate, right? So those two things are key giveaway. Oh, big nuclei in the center of the cell, intercalated discs, boom, cardiac muscle tissue, okay? So, and you can see on the outside covering again that sarcolemma or plasma membrane is going to be the endomysium, okay? Now, endomysium will be a connective tissue that connects, again, one cardiac muscle cell to the next, okay? And again, we talked about the intercalated discs, and you can see, we won't mark blood vessels, but there are blood vessels obviously supplying nutrients and oxygen to that tissue. Okay, so that is the model of cardiac muscle cell. Now, this model is smooth muscle. Easy peasy, right? Because look, spindle shaped right here, really large central nuclei, no striations, boom, smooth muscle, okay? And then you can see here in between, surrounding each of those individual smooth muscle cells is going to be the endomysium, okay? And we mark the fibroblast nuclei because endomysium is, again, a connected tissue, so it does have fibroblast nuclei like your, again, loose connective tissues do. Okay, so then we have skeletal muscle fiber. But again, if you just put nuclei, we're not going to mark the endomysium nuclei. We'd mark the nuclei on the smooth muscle cell, okay? So don't worry about that. And skeletal muscle, the skeletal muscle model. Okay, so again, you're going to ask yourself, is it striated? Oh, yep, it's striated. So now, is it skeletal or cardiac, right? Oh, are there intercalated discs? No, no intercalated discs. Oh, does it have peripheral nuclei? It sure does. Okay. So then you automatically know it is a skeletal muscle cell, okay, or skeletal myofiber. Okay. So again, you can see it marks the striations. You can see it's also going to have the endomysium surrounding that sarcolemma. The sarcolemma here, you can actually see the sarcolemma or the plasma membrane. It's going to be this stuff again covering, right? This individual myofiber that's, or holding it together. That's the sarcolemma or plasma membrane. And then this gray stuff in between is going to be the endomysium connecting one skeletal myofiber to the next. Okay. So hopefully that will all make sense for you now. So that is the end. Oops. As I accidentally zoomed here, that is the end of those muscle models. That's it. Okay. So then let's go back here and let's go on to nervous tissue. You're like, oh my gosh, this is already too long. Like, how are we going to remember this? You're going to love nervous tissue because nervous tissue is we're saying very basic, okay? And it's hard to get a good prep of nervous tissue. So you're only going to be looking at a multipolar neuron slide, okay? And so I will show you what this looks like in this first slide here. So you are only going to be looking at this first slide. Don't get into the neuromuscular junction and all of that because all of that is coming when we get to the muscular system and the nervous system. So again, ignore that. Only look at this first slide, okay? So when we look at nervous tissue, these cells are going to look very different because their purpose, right, is to basically pick up messages, right? And then pass those messages on. So you're going to have in your multipolar, here's your actual multipolar neuron. And here's going to be the nucleus, and then the nucleolus is really stained really dark in this. And then what you're going to see is it's going to have all of these little things that eventually you'll learn are called dendrites. And then one of those is probably an axon, but you can't tell which. And so inside those dendrites are going to be kind of taking in the messages and passing them along as they connect to other neurons. And then they'll pass it along through the axon. It's way more than you need to know right now. But just to give you an idea, that's why these neurons look so branched, just because they're taking in messages and passing them along. Okay. So you're going to see that they have these little dark fibers called neurofibrils. So myo again means muscle. So myofibrils were the fibers inside the myofibers, the muscle cells. And so neurofibrils are going to be the fibers inside a neuron. So again, just you'll have to learn that term. Myo means muscle. Myo means muscle. Neuro again means nervous. So for the nervous system. So pretty easy there. And then the only other thing that you're going to have to know is that surrounding this neuron are the support cells called the neuroglia. And the neuroglia are these lovely little tiny cells that are basically there to support the neuron, to feed it, to protect it, to keep it safe, to hold it in place. Because the neuron is far too busy. You know, neurons in your brain are constantly sending messages back and forth. They are far too busy to take care of themselves. So they have all these lovely helper cells called the neuroglial cells to take care of them. And so these neuroglial cells are going to be these little tiny, you can only see the nuclei of them, these little tiny cells that are surrounding that neuron. Okay. And that's all you have to know. You just the multipolar neuron. There's the nucleus and the nucleolus. If you put nucleus, I would still accept that as long as you knew it was the nucleus inside that multipolar neuron. And then you could see these, as long as you know that these are the neuroglia helper cells. So neuroglia helper cells. And again, learning these terms now will help you when we get to the nervous system, because we're going to talk a lot about neuroglial cells because there are lots of different types of neuroglial cells. So hopefully that will help you when we get there. But that's all you have to know. Really easy, right? Pretty basic. Not a lot that you have to know for the nervous system because we get into that later on. So please keep in mind that you do have soft chalk labeling activities. Please do those. They're super helpful. And then you also have StudyMate flashcards. So make sure you do those. They're really helpful. And then if you go back to, return back to histology, again, don't forget to take the practice quiz down here, okay? And I did have a question of the practice quizzes. Please remember the practice quizzes are not graded. They're just to help keep you on track. I did put due dates on them, but there's no late time to take them because again, they're not being submitted for grade, but I only put due dates on there to kind of give you an idea of when you should be ready to take a quiz on that topic. So again, please use those practice quizzes. They should really help you out. And then also remember that pretty soon here, and I will send out a notification. I try not to open it too early because if I do open that practical too early, people will think, oh, well, I'll just study that. But that practice practical, and I put this here, this practice practical is for practice only. It is not meant to substitute studying. There may be things on the practical that are not tested in there. It's just to give you an idea of what types of things you can see, right? But you might have a question on, you know, cardiac muscle tissue in this practice practical, and then it might be on skeletal muscle tissue in the actual practical. So please, again, don't think that you can study only that practice practical. Study for the practice practical like you were studying for the practical, and then take it and see how you do, okay? Also keep in mind that I am not grading those practice practicals, so Canvas is not smart enough to know that you capitalized the answer and maybe we didn't put the answer in capitalized, okay? So you would still get that correct, and I will be hand grading each of your practicals. I will give you points back for things like that, but Canvas isn't smart enough, okay? So if you ever have questions and something was marked wrong on the practice practical and you're like, would that be marked wrong? Like someone asked me, they had put lacuna instead of lacuni, or lacuni, the plural, instead of lacuna. And again, if you did that in a practical, you got it right. But it may mark it wrong on the practice practical. So again, I will be adding back in those points. So don't sweat that, okay? Again, if you have any questions, please let me know, and I would be happy to set up some time to do histology. As you can see, I love histology. I know I'm sick and wrong that I love histology, but it's always fascinating and interesting to me. So, okay, hopefully that will help you out and help you get started, and have a great week with lab.